So good evening everyone. Welcome to my YouTube class. So how are you all? Hope you are all doing good. Super. Welcome everyone to my YouTube session. So waiting for the students to join. So there is a plus program running in Unacademy. So in this you can get complete syllabus get covered, tips and tricks by top educators, you can get it, topic wise practice questions, full length marks, all just at the rate of very nominal price 2250. For that you can use the code of GHOUSELIVE, GHOUSELIVE, so that you can get 10% off in plus subscription. You can get 10% off in plus subscription. So above the different plans, you can also have the different plans. One is 1 month plan, as already told to you. Then 6 months plan we have, and 12 months plan. So as I recommend you KCET, you can use this 6 months plan. So now you may get it out, sir, what I will get in this comprehensive batch. So you can get premium content at very affordable price, structured live courses and dedicated live doubt clearing sessions, quizzes at regular intervals for self-analysis, batches for complete preparation. So every month you will get new batches. So there will be a test series for DPPs, full length marks, etc. with analysis. So once you get single subscription, you will get unlimited access. This is the beauty part. If you are one educator, you can listen all the educators who are teaching in the Unacademy platform. So hope you are all doing good. Welcome to everyone. So waiting for the students to join. So very good evening everyone. So very good evening everyone. So good evening everyone. How are you all? Welcome to my class. So waiting for the students. So welcome everyone. So today our topic is guys, yes, as you know, what is our topic? You already seen, right? Yes, in thumbnail picture, right? Selection of transformed cells. Selection of transformed cells. Selection of transformed cells. This is topic for the day, guys. Okay, so let me start. Hope everyone is safe. So if you get any doubt, you can post on the comment box, live chat box, so that we can discuss all your queries. Okay, so now students, before going to explain about that, so what we have to do is, we have to understand first of all, what is PBR 322. If you understand PBR 322, then we can think about and we can learn about the selection of transformed cells. Now students, let me talk about how PBR 322 is going to be there. So students, this is a plasmid. So please forgive me here. Just I am drawing without using any tool. I am drawing. Hope you understand. So you have two selectable markers. one is right side, so this is calling it as tetar, this is calling it as tetar. Other side, this is calling it as AMPR, the other side is calling it as what? AMPR. So this is tetracycline resistant gene, this is ampicillin resistant gene. And now you will have ORI and you have ROP. As you all knows, what is ROP, students? As you all knows, what is ROP? ROP nothing but what? Region of protein coding. ROP is what? Region of protein coding. Whereas ORI means what here? Origin of replication. Now you may get what is AMP, R and what is tetar. Means these are antibiotic resistant genes. Okay, antibiotic resistant gene. Okay, now before going to know that, you have to know the restriction enzyme sites. So which all enzymes act on which part. So here at this site, echo R1 will have the restriction site, CLAV1 here, and this is HIND3. On tetar, you will have two enzymes. One is BAMH1 and SAL1. And this side, you know, we have PBU1 and PST1, which have restriction sites here. Okay, and then here ROP is there. So at ROP we have PBU2. At ROP we have PBU2. Okay, at ROP we have PBU2. What is PBU2? You know, Protease vulgaris 2. What is PBU2, students? Protease vulgaris 2. Now, the main important point what you have to keep in mind is, yes, so now PBr322 is there, yes, students. So in this PBr322, please draw a diagram now, everyone. So waiting for your doubts here. So students, you can post the comments on the chat box. Yes. So now this is PBr322. So now what you have to know is, suppose our insert. So insert is nothing but what here now? Insert is nothing but what it is? Desired gene. Insert is nothing but what, students? Desired gene. So when you insert, okay, depending upon the restriction enzyme which you use, that particular antibiotic restriction gene will become inactivated. So I repeat again, I will take another slide now. Say I used. So suppose if the restriction enzyme is, yes, if the restriction enzyme is BAMH1, if the restriction enzyme is what? BAMH1, then how can you select? So our topic today is what? Selection of transformed cells, right? Selection of which cells? Transformed means transformation happen or not, right? There's a topic. Suppose you know if BAMH1 acts, so first thing you have to understand is what? Where BAMH1 having restriction site? Tell me. Where BAMH1 having restriction site? Please guys, post in your comment box now. Where BAMH1 having restriction site? Please guys, fatafat, do it now. Post. Fatafat, no waiting. Yes, you know, tetar, right? It is having a tetar means, okay, now it is going to cleave which part? tetracyclistin part. So students, now what I have to do? I have to culture the cells. Culture the cells in antibiotic resistant media. Simple. If I want to know whether transformed cell is there or non-transformed cell is there, then what I have to do, guys, here? Very simple. I have to grow in the antibiotic medium. So the point which I am going to tell here is golden points now for that. So if you know this point, no, in your lifetime you never forget about the this selection. Okay, students. So let's focus on this. So students, if desired gene is present, nothing but insert is present, then what happen? Selectable marker is absent. Selectable marker is up because that enzyme act on that, right? Yes. Then if desired gene is present, means present. Okay. Then second case is, this is case 1. Then next, what will be the case 2, guys? If desired gene is not there, if desired gene nothing but what it is? Insert, right? Insert is not there means what? Selectable marker is active. Selectable marker is present. Selectable marker is present. So this is the key note point you have to keep in your mind, guys. Okay, if desired gene insert is present. Okay, so let me draw again here. I'm searching for the tools to draw ASS. So another rough diagram doing here. Okay, you see here. This is the tetar. Yes, tetar. So BAM H1 acts here means if BAM H1 cleaves this, then our insert gene is introduced here. If our insert gene is there means, present means selectable marker is absent. Tetracycline is resistant gene is not there. Tetracycline resistant gene is not there. So now, guys, if insert is not present means what? Here? Tell me now, quick. If insert is not there, means tetar is active. Yes. Guys, you please mention in the chat box. If you know this, then next topic is going to become very simple to understand the concepts. Yes, guys, mention your comment box now. Are you all listening? How was the day today? Guys, you can catch me on Unacademy KCET app. You can follow me there. So every day we are coming with mock KCET exams. So if this is the first time of you in YouTube, you can please follow me on KCET app. So Unacademy KCET platform, you can catch me. You can follow me there also, so that you can get all the notifications which you'll get. Yes, guys, clear now? Fatafat, tell me now. Fatafat. Waiting for your answers in the comment box. Fast. Yes. Now I'm going to take another screen. Wait. Yes. Now. So I want to know whether selection happened or not. Selection procedure now. What should I do now? First point. Yes. What should I do now? You have to keep. Yes. Selection procedure means what? We have to grow in antibiotic media. We have to grow in which media, guys? Antibiotic media. Antibiotics means what here? What are the antibiotics? Tell me, guys, what are the antibiotics? Fatafat. Yes, tetracycline and ampicillin. Tetracycline. Second one is what, students? So tetracycline and ampicillin both are what students? These are tetracycline, ampicillin, both are which medias, you know? Yes, antibiotics. So now you have to keep one point in your view. So what is that point you have to keep in your mind? Let me know quick. Restriction enzyme. Which restriction enzyme you used? That is one of the important point. Restriction enzyme used. Which is the restriction enzyme used here? I told you already. What is the restriction enzyme? BAM-H1. Yes, where it has restriction site? TET-R. Now students, when I grow in the media, then you say here, this is 2 Petri plates I have taken. So one is having tetracycline, the other one is having ampicillin. The other one is having what now? Ampicillin, tetracycline ampicillin. Now tell me here, fadafat now. See, the red dots are growth. Bacteria colonies keep. Like that. Grow. Here no growth. No growth. Now tell me here which is transformed, which is not transformed. Which is transformed? Hi Venkatesh. How are you? Tell me now, fadafat. No growth now. Yes. So that growth is happening means what? See, this is the case 1. Assume this is the case 1. Or case 2. Just for example to you. So growth is happening. So growth is happening. Means growth is occurring. Okay, growth is present. Growth is present means which gene is active? You have to think. Which gene is active? Which gene is active? Yes, which gene is active now? TET-R gene is active. If TET-R is active, which will be not there? Insert is absent. Insert is absent means it is a transform or non-transform. It is absolutely non-transformed. Absolutely it is what students? Non-transformed. Absolutely it is non-transformed. Absolutely it is non-transformed. Got it students now? So growth is happening, growth is occurring means what? TET is there, TET is active because it's an antibiotic. Basically bacteria cannot grow in antibiotic. Basically bacteria cannot grow. Bacteria cannot grow in antibiotics. Bacteria cannot grow in antibiotics. It is growing means TET-R is active. TET-R is active means yes, this is the point. Please make a note here. Fadafat. Please mention your doubts in comment box. Now, no growth. Tell me here for no growth, what will be the reason? For no growth, tell me what will be the reason? No growth means, okay, now a small point you have to think is, okay, small point you have to make a correction here. Okay, we can do anything else, no problem. Okay, assume this is only tetracycline. Okay. Okay, now this is transformed. Okay, this is transformed. Just students, for the sake of your understanding, just I'm telling. Okay, tetracycline, now it is transformed, means no growth. Let me talk about how no growth will come. Take this fresh slide here. So no growth. No growth means what? No growth means what? Antibiotic resistant gene is antibiotic resistant gene is present or absent? Absent. Because of that only no growth. Antibiotic resistant gene is absent. Antibiotic resistant gene is absent. Because of that only. Okay, which gene is absent here? See, because we used which enzyme? BAM H1 na? We used which enzyme, guys? BAM H1. When you are using the enzyme of BAM H1, where it acts? It acts on TETR. So TETR is inactive now. TETR is inactive. So TETR is inactive. When TETR is inactive, which is active now? Insert is present. Insert is present. What is our insert? Desired gene. What is our insert, students? Desired gene. So antibody resistant gene is absent means our insert is present. Insert is present. So insert means we can mention in the bracket also desired gene. You can mention the bracket is what, guys? Desired genes. So if desired gene is present means, is it transformed or transformed? Non-transformed. It is transformed cells. Transformed cells. Okay. Simple. Like this you can grow. You can get it now. So students, once again I will draw the diagram of PBr322. Very simple rough diagram. Okay. So here we use the enzyme of BAM H1. When you use the enzyme of BAM H1, it acts here. TETR. If TETR is active, if here simple, that's what I told you. TETR is active means it can grow. TETR is inactive. TETR is inactive. Hello guys, how are you all? If TETR is active, if TETR is inactive, if TETR is active means it will grow. If it is inactive means it cannot grow, can't grow. So it is cannot grow means what happened, guys? Tell me quick, fatafat. For this what happened? Tell me. It is able to grow means what? TETR is active means insert is absent. Insert is absent. TETR is inactive. TETR is inactive means insert is present. Insert is present. Insert is present. So if insert is absent means what? So can I call, say this is the row number 1, row number 2. Now guys, tell me here which is the main thing here, which is transformed, which is non-transformed. This is non-transformed. Why? Because why it is a non-transformed, students? Insert is absent means TETR is active. If TETR is active means no our desired gene. Then case 2 is what? TETR is inactive means that place we added our gene. So that's why it can't grow, because resistant gene is inactive. So if resistant gene means antibiotic resistant gene is inactive. If antibiotic resistant gene is inactive means insert is present. If insert is present, then it is called as non-transformed or transformed? Transformed. Yes. So this can also be called as replica plating technique. So we can use the replica plating. Okay. 2 plates we have to keep 2 plates, right? In the same way, I have told you, suppose same thing. Let me take same thing. You use the, suppose our restriction enzyme is BAM H1 only, but you are growing in which media? Ampicillin media. Keep in mind, ampicillin media. Okay, now just recollect guys. Now take the plasmid structure. Okay, every time you have to remember. Okay, this is TETR, this is AMPR, TETR and AMPR, right? So BAM H1 is acting on where? TETR only. Okay, BAM1 do not have restriction site. do not have restriction site at AMPR, right? So now tell me here, you have to grow in which media now? Amphiphilic media. Then you'll get two cases, transformed cells and non-transformed cell. You will get two now, transformed and non-transformed. Now tell me here, transformed cell it will grow, non-transformed cells don't grow. What it is? Tell me guys. Students, are you all on the line? Venkatesh is on the line. Please. Please, fatafat. Post in your comment box. Fast, fast. No time here, already 10 now. Yes, now students, whether in the case of transformed cells also will grow, non-transformed cells also will grow. Why? BAM-H1 is not acting. See, BAM-H1 do not have restriction site at AMPR. So, whether it is a transformed or whether it is a non-transformed, okay, it do not show any effect. Why? Because our enzyme is what? What is our enzyme is here? BAM-H1. BAM-H1. So, like these students, you can select the transformed cells. You can select the transformed cells. Okay, then here one definition you have to understand here, insertional inactivation mechanism. What do you mean by insertional inactivation mechanism? When you insert a gene, when you insert a gene, okay, one of the selectable marker is going to become inactive. One of the selectable marker become inactive. One of the selectable marker become inactive. Nothing but one insertion activation mechanism. So in insertion inactivation mechanism, one of the marker is going to become inactive. So by using this mechanism, we can able to identify transformed cells as well as non-transformed cells, as well as transformed and non-transformed. So using selectable markers only we can able to identify by using which mechanism? Insertional inactivation mechanism, students. So hope you all got clarity here. Any doubt, students? Please, fatafat. You can mention the comment box on this. I'll show you all the slides once again. Observe the slides. This is a PBR 322 slide. Students, if you are first time in my class, you can follow me on Unacademy KCET app. You will get more marks, more classes, everything. So you can share the video link of YouTube to your friends also. So they'll get benefited with these YouTube classes. Post in comment box. Guys, are you all understand now? Tell me here. This is PBR 322. So insert is nothing but SI gene. So it all depends. Selection depends on restriction enzyme used. Say if the restriction enzyme is what here? PBR 322. Okay, restriction enzyme is BAM-H1. If you use BAM-H1, where it acts tetar, it acts. So that part will get inactive. If that part become inactive means, if desired gene is present means selectable marker is absent. So this point you have to keep in mind. Only thing you have to culture in the resistant genes. So suppose you cultured See, you have taken, you cultured in antibiotics. Say tetracycline you cultured. Okay, tetracycline you cultured. This tetracycline, this is tetracycline. So non-transformed is growing, you see. If they are growing means that resistant gene is active. That resistant gene is active means our disease gene is absent. So it is non-transformed. Is no growth, is not growing. Not growing means what? That tetracycline resistant gene is not there. That tetracycline resistant gene is not there. If that gene is not there, is a transformed. So you can keep focus here. Growth is present means which gene is active. Gene is active means insert is absent. So students, it takes some time to you also. So simple logic. Insert is present means 3 points you have to keep in mind. Only 3 points. What are the 3 points here? Where restriction site is having. After that, which enzyme you are using, that only it is. Then insert is present means cell table marker is inactive. If cell table marker is inactive means it cannot grow in that media. It cannot able to grow that media means our gene is present. Our gene is present means transformed cells. Now students, tell me here whether you all understand the class or not. Fatafat. Clear na? So in your lifetime again you should not forget this selection. Okay students, hope you all understand the concept here. So my name is Syed Gauss. So I am the faculty member at Unacademy. So I am taking plus classes also. For that you can join me in the plus as well as the free classes also. So thank you all guys. So very good evening everyone. So before good evening, now I have to say a good night. So good night to all the students. So thank you, bye bye. Tomorrow we'll meet the same time again in the YouTube platform.